Subscribe to our channel by clicking the subscribe button. Click the bell button and enjoy the latest uploads from our channel.
Question number 15. Write any 4 ways to introduce a desired DNA segment into a bacterial cell in recombinant technology experiment. क्या हो सकता है? Desired DNA segment is inserted into cloning vector. Cloning vector में insert करेंगे desired DNA segment को. Bacterial cells are made to take it up after making them competent by treating them with specific concentration of divalent cation. Divalent cation may be calcium. Or second method is micro injection. Recombinant DNA directly inject करेंगे into the nucleus of an animal cell. That is micro injection. Biolistics और gene gun cells bombard करेंगे with high velocity microparticles of gold or tungsten coated with DNA. Disarmed pathogen vectors infect the cell, transfer the recombinant DNA into the host. ये है 4 ways to introduce a desired DNA segment into a bacterial cell in recombinant technology experiments. This was question number 15.